Hello everyone, we're going to continue on by recapping the sample exam question or test question, and this was reaction 2 on the previous lecture. So here we have our reaction. We have one molecule, AB, and it is being broken down into two molecules, A plus B. Those are two individual molecules. So we're going from one molecule now to two. So you should see right off the bat that we're increasing the disorder within the system. We're going from one molecule to two. So therefore, we could assume that we're increasing entropy. Okay, so let's check out the parameters here. The first is that we need to calculate the change in enthalpy. So based on our calculation, we have H final minus H initial, which is equal to the numbers that we sub in. So the final enthalpy is 25 joules minus 6 joules, which was our initial enthalpy. And so the difference there is a plus of 19 joules. So from an enthalpy standpoint, the system is actually gaining energy. So when the system goes through this reaction, it is gaining 16 joules of energy. Okay, so we're gaining, we're in a positive enthalpy. Now let's look at entropy. So we need to calculate the delta or the change in entropy. So S final minus S initial, we sub in the numbers that were given above. So 45 joules per degree Kelvin minus 34 joules per degree Kelvin is equal to a plus of 11 joules per degree Kelvin. So from an entropy standpoint, the system is actually increasing disorder, increasing entropy, which we already knew this because if we look at the sample equation, we go from one molecule to two. And when we increase the number of molecules within the system, we're increasing disorder. It's like digestion, where we go from one apple and we break it down through the process of digestion into many molecules. Well, the system is increasing entropy, and therefore we have a positive number. But remember, this is very important, that there's an inverse relationship between entropy and energy, such that when we increase entropy, we're actually losing energy within the system. Okay? So in this case, from an entropy standpoint, the system is losing energy. So the question becomes, okay, so what's the overall system doing in regard to energy? Is the system gaining or losing energy? So now let's talk about how we would determine what is happening from an energy standpoint for this reaction. Okay, so we have to identify what we know. One thing we do know is the temperature. So this is highlighted in green here. The temperature of this reaction, it's occurring at 20 degrees Celsius. That's really important because temperature will determine whether the reaction is favorable or not. Okay, it's an important factor that determines whether a reaction is favorable or not. So if we remember our laws, which are at the top of this slide and highlighted in yellow. So for an exergonic reaction that releases energy, the change in enthalpy has to be less than temperature times the change in entropy. And then if a reaction requires energy, so it's endergonic, the change in enthalpy will be greater than temperature times the change in entropy. So we can calculate this, and we're going to do this on the next slide. So now we're going to determine how we would solve this with Gibbs free energy. So using the two variables that we calculated on the previous two slides. So the change in enthalpy is plus 19 joules, and the change in entropy is plus 11 joules per degree Kelvin. And now we have to calculate the temperature in degrees Kelvin. So we know what it is in degrees Celsius, but we have to convert that to degrees Kelvin. So that's our first step. We see number 1 here on the slide. It says convert degrees Celsius to degrees Kelvin. So this is equal to 20, so that's what we know the temperature is. 20 degrees Celsius plus 273 is the constant that's needed to convert degrees Celsius to degrees Kelvin. So in this case, our temperature is 20 degrees Celsius plus 273 equals 293 degrees Kelvin. Okay, so you can add that to the slide. So when we convert degrees Celsius to degrees Kelvin, it's the temperature plus 273, which is equal to 293 degrees Celsius. Okay, we did that. We checked number 1. Number 2 on here is calculate what the total is when we multiply temperature that we just calculated, 293 degrees Kelvin, times the change in entropy, which is 11 joules per degree Kelvin. So we times 293 degrees Celsius times 11 joules per degree Kelvin, which is equal to 3,223 joules. Okay, so you write that down. It's 293 degrees Kelvin times 11 joules per degree Kelvin equals 3,223 joules. Now we need to go back to our rules to determine what is going on with this reaction, whether it's exergonic or endergonic. So if we know the change in enthalpy is 19 joules, is it less than 3,223? Yes, it is. So our first rule is actually true. So in this case, the reaction would be exergonic, okay? Because 19 joules is less than 3,223 joules that we calculated for temperature times entropy. So in this case, our rule indicates that this reaction is exergonic. So this is how you would solve for a reaction when we have two systems that are positive. We have a positive gain in entropy and we have a positive gain in enthalpy. And we don't know what is happening with the system. So what we have to determine the degrees Celsius, convert to temperature, and use these two rules to determine whether the reaction is exergonic or endergonic. Based on the previous slide, we just determined that this reaction is exergonic because delta H, which is 19 joules, is less than the calculated temperature times entropy, which is 3,223 joules. So therefore, based on our rule system, this would be an exergonic reaction, where the change in enthalpy is less than temperature times the change in entropy. So you may be asking why we do care about delta G in biochemistry. Why is this important? And I certainly, when I was a student, I asked this question. And it's because the change in Gibbs free energy determines whether a reaction is energetically favorable, and therefore whether the reaction is spontaneous or non-spontaneous within the body. And we want reactions that are spontaneous to be ones that release energy, for which then our body can use for muscle contraction and different functions within the body. So for a reaction where the delta G is negative, this is favorable. This means that we're releasing energy by the reaction. And this reaction is therefore termed exergonic. And the reaction will occur spontaneously in the body. We do not have to wait for it to happen. It will happen spontaneously. And these exergonic reactions release energy within the system, within our body. Remember, energy is neither created or destroyed. It changes forms. And so from this breakdown, from this reaction, energy is released. And then what our body can do is use it for muscle contraction, for sodium-potassium pumps, for all the cellular maintenance that we need. Now let's look at some reactions to see the importance of delta G in biochemistry. So on the right, we see two reactions. One is labeled number 1, and the second reaction is labeled number 2. And these are the first two steps of glycolysis, which is the breakdown of glucose. So we eat food that has carbohydrates in it. They get broken down through the process of digestion and enter the blood as glucose. Glucose has to circulate around the body until it's taken up by the cells in the form of glucose, and now has to be broken down so that we can create energy, or energy currency, which is ATP. So these are the first two reactions. We have glucose being converted into glucose 6-phosphate, which is reaction 1. And reaction 2 is glucose 6-phosphate being converted into fructose 6-phosphate. And we'll learn more about this in the coming weeks when we talk about glycolysis. What I want to draw your attention to right now is the directional arrows. So in reaction 1, we have a red arrow that indicates that the reaction is only going in one direction. And in this case, it's called an irreversible reaction. So it's called a unidirectional arrow that is only going in one direction. Uni meaning one direction arrow. So this indicates that that first reaction is irreversible. It only operates in one direction. And as you can see, the delta G value, minus 16.7, is highly exergonic. Therefore, this reaction happens spontaneously, it's irreversible, and therefore reaction 1 is a spontaneous and irreversible reaction. Okay, that's important. Any exergonic reaction is going to be irreversible and spontaneous. The second reaction, as you can see, the arrow is different. It has a bidirectional arrow. The arrows point in two different directions. It can go in the forward direction or down, or it can go in the backward direction or up. So this indicates a reversible reaction, that they operate in both directions. So we can go from glucose 6-phosphate to fructose 6-phosphate, and then from fructose 6-phosphate back to glucose 6-phosphate. Okay, so this reaction, as you can see, the delta G is positive. We have positive 1.7. So when delta G is positive, then it's a non-spontaneous reaction. The arrows indicate that it's reversible. So reaction 2 is considered a non-spontaneous reversible reaction. One more point about this slide is I want to draw your attention to reaction number 1. So this is glucose being converted to glucose 6-phosphate. And as you can see, something else is being added to this reaction, and it is ATP. So we actually have our energy is being put in to this reaction. Well, why do we need that to happen? Well, we're going to talk about that on the next slide. But what I want you to realize is that this first reaction of glycolysis is very important. We need to convert glucose into a bigger molecule called glucose 6-phosphate so it doesn't move back into the blood. So as I mentioned, through the process of digestion, we break down carbohydrate to the smallest monomer. In this case, it's mostly glucose. Glucose travels in the blood and it gets taken up by our cells. But we want the glucose to stay within our cell. So as soon as glucose enters our cells, it goes through the spontaneous, irreversible reaction to get converted into a bigger molecule so it doesn't move back through the channels and back into the blood. We want to trap glucose within the blood, so we make it a bigger molecule of glucose 6-phosphate. So we need this reaction to be exergonic and happens spontaneously. So we're going to talk about why we add ATP to this reaction on the next slide. So now I want to talk to you about that first reaction of glucose oxidation, where we have glucose within the cell and we need to trap it in the cell by making it a bigger molecule. So we go from glucose to glucose 6-phosphate. Well, initially, if we were just to add, if we look at this chemical equation up above, glucose plus an inorganic phosphate plus a hydrogen ion leads to glucose 6-phosphate plus water. If we consider this reaction of adding a phosphate, this reaction is actually consuming energy, and therefore the delta G would be 13.8 kilojoules per mole. So it would be an endergonic reaction, and therefore it would happen non-spontaneously. But yet we need this reaction to happen spontaneously and hopefully not consume this much energy. So how do we change this reaction to make it spontaneous in life? How do we do this? So in nature, initially, this reaction does not occur on its own, but we need to be spontaneous because we need to trap glucose within the cell in order to start glycolysis in the process of making energy. So we need glucose to be converted into a different molecular structure so it is trapped within the cell. So bear with me for a minute and I'll continue on talking about this on the next slide. Now we're going to talk about the solution to this problem when we have a highly endergonic reaction like on the previous slide, but we need that first step of glycolysis to be exergonic. What we can do is we can combine reactions and we can combine or add ATP hydrolysis, the breakdown of ATP, to make a reaction energetically favorable or to convert it from being endergonic to exergonic. So how do we do this? Well, ATP itself has these high energy phosphate bonds. And every time we break ATP down to ADP to AMP, we release energy. So you can see in the optic on the right, if we look at the very bottom that's circled, we have ATP. And when we break it down to ADP plus an inorganic phosphate, we actually release minus 30.5 kilojoules per mole of energy. So that's a lot of energy. That's why these bonds are called high energy phosphate bonds. And the breakdown of ATP yields a lot of energy. Then the reaction above that, where we have adenosine diphosphate, ADP, being broken down through hydrolysis to AMP plus an inorganic phosphate, we release another 30.5 kilojoules per mole of energy. Then when we break down AMP, adenosine monophosphate, that only has one phosphate, to adenosine and phosphate, we release 14.2 kilojoules per mole. This is why ATP is highly energetic. Like the breakdown of the high energy phosphate bonds releases a lot of energy. So when we add ATP to certain reactions, we can actually change the reaction from being endergonic to exergonic. So this is called ATP coupling. And it makes a reaction energetically favorable and spontaneous. And this is exactly what happens with the first step of glycolysis when we see ATP being added to the reaction. So in biochemistry, whenever you see ATP going into a reaction, we think it's weird because we're actually consuming energy. Well, no, what's happening is we're breaking down ATP and using the energy to make a reaction favorable and spontaneous. So here's our solution as we see it in regards to combining reactions. So we have our reaction 1 from 2 slides ago, which is the phosphorylation of the glucose reaction, where we have glucose combining with an inorganic phosphate to make glucose 6-phosphate plus water. And if we draw your attention to the delta G of this first reaction, reaction 1, R1, it is plus 13.8 kilojoules per mole. Highly endergonic, and therefore it would be non-spontaneous. But this reaction is the first step of glycolysis, and we need to convert it to a spontaneous exergonic reaction. So what we do is we combine it with ATP hydrolysis. So if we look at reaction 2, we have water plus ATP leads to ADP plus an inorganic phosphate. So we go from one molecule, which is ATP, we add water and we break it into 2 molecules, which is ADP and an inorganic phosphate. And look at the delta G of reaction 2. We see it's highly exergonic, minus 30.5 kilojoules per mole. One of the most exergonic reactions of the entire body is the breakdown of ATP. So we combine these 2 reactions together, which I'll show you on the next slide. So what's the benefit about doing this? Well, this is called ATP coupling. We add ATP to a reaction to make the reaction exergonic. And so it involves the removal of a high energy phosphate from ATP. So we're removing a phosphate, we're dephosphorylating ATP. And it provides a means to change the reaction from being endergonic to exergonic, and therefore favorable and spontaneous within the body. Now, when we combine these 2 reactions, we can calculate the overall delta G. So the delta G of reaction 1 is plus 13.8 kilojoules per mole. And then we add in the minus 30.5 kilojoules per mole of reaction 2. And this makes a delta G of minus 16.7 kilojoules per mole. So we're combining these 2 reactions. So the way in which we do this, I'll show you in class because I need to write it out, but this is called a coupled reaction, where you're adding together one reaction plus ATP hydrolysis to convert it to an exergonic reaction. And therefore it's spontaneous and happens energetically favorably within the body. And this is the first step of glycolysis. So whenever you see ATP being added to a reaction in the body, we know that we had to convert that reaction to become energetically favorable. So that reaction initially was very endergonic, but it's an important reaction that we need to happen spontaneously and be exergonic. So that's where whenever you see ATP being added to a reaction, it looks like it's energy consuming, but actually it's becoming exergonic. So it's very important to be able to identify that. You'll see it when we talk about glycolysis, as well as other processes involving carbohydrate metabolism. Here's another example that I would like you to work through firstly on your own. So you can pause this video and try to figure it out on your own. But it's another example of ATP coupling. Okay, so let's look at reaction 1. You don't need to know all the details of this. What we do know is that water is being added to the reaction to form two products. So this is a hydrolysis reaction. We have one large molecule with a phosphate, water is being added, and now we have two molecules. We do not know what the delta G is for reaction 1. That's our unknown and what we want to try to solve for. Reaction 2 is ADP. We're adding an inorganic phosphate to it. We're actually forming ATP and we're removing water. Okay, so it is a condensation reaction where we're removing water. We're going from two molecules to one larger molecule, which is ATP, with the removal of water. So this is a highly endergonic reaction. It's consuming energy. We're adding a high energy phosphate group to this reaction, and therefore it's consuming energy. It's highly endergonic. So the delta G of reaction 2 is plus 30.5 kilojoules per mole. That's a known. We know that. Okay, the overall reaction is, you can see it there, and then the overall reaction delta G is minus 31.4 kilojoules per mole. So based on our equation, where we calculate the overall delta G equals the delta G of reaction 1 plus the delta G of reaction 2. That's our equation, and we need to solve for the delta G of reaction 1. Okay, so I want you to try this on your own by rearranging the equation and putting in our known variables. Now, I hope you set aside some time and tried to figure out this equation on your own before looking at this slide. So remember, we're trying to solve for what the delta G of reaction 1 is. That we have two reactions that are combining together to form an overall reaction. So reaction 1 is a hydrolysis reaction where we go from one molecule to two molecules with the removal of water. Reaction 2 is a phosphorylation where we're adding a high energy phosphate to ADP to make ATP. Okay, so this is energy consuming. And then our overall reaction, we are actually producing ATP, and we want this reaction to be exergonic. We want it to be favorable. We want the creation of ATP to be favorable. But if we do it from ADP, adding an inorganic phosphate, it's highly unfavorable because it's a plus 30.5. It consumes a lot of energy. So we need to combine reactions to make the production of ATP a favorable and spontaneous process. So if you've rearranged your equation, you know that the minus 31.4 kilojoules per mole, which is the overall delta G, is on the left-hand side. And then we're trying to solve for the delta G of reaction 1. So we rearrange, we bring the 30.5 kilojoules per mole over to the other side, to the left-hand side, and we end up having a value of minus 61.9 kilojoules per mole. So by combining these two reactions, we make a reaction that we want to be spontaneous and energetically favorable, which is the creation of ATP in the body. We want that to be a favorable reaction. But in nature, it wouldn't be. So what the body does by biochemistry is we actually combine these reactions together and make the overall delta G value a negative, highly exergonic, and therefore this reaction will happen spontaneously. So now we know that the creation of ATP happens spontaneously within the body when it's combined with other reactions. Very few times, if any at all, that you'll see the ADP plus inorganic phosphate and the creation of ATP reaction happening on its own. You'll see it always in combination with another reaction to convert it to be energetically favorable. In summary, two things determine if a reaction is favorable. It's enthalpy, or the heat change of the reaction, and the entropy, so the change in disorder within the reaction. Exergonic reactions are considered to be spontaneous, mostly irreversible, and are the most favorable within the body. And this usually has a negative change in enthalpy and a positive change in entropy. Delta G combines both the change in entropy, and the change in enthalpy, along with temperature. Coupling reactions with certain things like ATP hydrolysis or ADP phosphorylation can make non-favorable reactions favorable. So remember that typically when ATP is added to a reaction, we're changing it from endergonic to exergonic. And when we're creating ATP, it's usually always combined with another reaction to make it favorable, so that we're always producing ATP within the body.